afraid we have to go start it now, but we are half empty. I hope the people will drop in. So it's my great pleasure to introduce you, dear Paul Orchard, who is from the United States and will give us a very inclusive talk about HCST. Paul, we just decided that you will introduce yourself.
That's
you.
fine. I'll give maybe a little bit of background. So I'm a pediatrician, trained in oncology and bone marrow transplantation, and at least in our institution, people tend to gravitate to either doing just transplantation or doing oncology, and I'm a transplanter. And then within the transplant side of things, I pretty much specialized in doing the rare inherited diseases as opposed to leukemias and other things. So I've been doing this for quite a period of time, and I'd like to touch on a number of different things today, but hopefully we'll have a few minutes at the end too if we can ask, if there's questions to be asked. So, just in terms of disclosures, we've had clinical trials with a number of different groups that have helped support those and also have helped us with our research. So a few things I'd like to touch on today, and talk a little bit about background for why we do transplant for these kinds of diseases, and talk a little bit about the experience for some of the MPS disorders, and talk a fair amount about Hurler, just because we have more experience with that, and transplant's really the standard of care for the patients with Hurler. Then we're going to talk a little bit about where things are going, because I think there's a lot of things that are changing in the field. So maybe to start, I mean, why would it make sense to use transplant for these kinds of diseases? So this is actually some information from Liz Neufeld. So Liz Neufeld is a researcher. She's actually here at the meeting. I just saw her a little bit ago. And she did this really important work back, this is from the late 60s, and showed that if you have cells from Hurler patients or Hunter patients, they accumulate a lot of this storage material. But if you mix them together in culture with normal cells, that accumulation of storage material really goes away. So this is what people call cross-correction. So you have normal cells that can correct cells that are accumulating, that are enzyme deficient cells, and at that point they didn't know that what these enzymes really were, but they knew that normal cells could help correct them. So this is really the basis for why people thought that transplant might make sense. If you can have a normal cell that's producing enzyme, excuse me, next to a cell that doesn't have enzyme, the cell that's making enzyme can help out the cell that's deficient. I'm just showing this to say that, so a lot of the transplant-related experiments started in the 80s and took off from there, but obviously now that all these genes have been identified and cloned and there's pharmaceutical products available, there's a number of different opportunities to treat these patients besides transplant now. So what diseases then would make sense to consider transplant? So this is a pie chart with virtually all the lysosomal diseases, or at least the vast majority of them. And I divided it into the diseases that don't really affect the brain or the nervous system, which are in blue, and the ones that do, and those are in red. So, and these are the diseases that we primarily are transplanting now. So the point is that most of the disorders that don't really affect the brain are being treated in other ways, enzyme therapy, for instance. But the diseases that can affect the brain and have cognitive problems, those we're still considering using transplant for. So why would transplant help the brain out when enzyme therapy doesn't? So there's a particular cell called the microglial cell that comes actually from blood cells. And so if one does a transplant and establishes normal cells that are in the bloodstream, that those cells can become a number of different types of cells, one of which is the microglia. So over time after transplant, some of these cells from the donor make their way into the brain and we think provide some enzyme there. So the blood-brain barrier keeps out intravenous enzyme if it's administered, but in the situation of a transplant, you can actually get cells into the brain and provide some enzyme. Now, there's a lot of things we don't understand as well as we would like. How quickly does this actually happen? What proportion of the cells actually get replaced? Are there things that we can do that can manipulate getting more cells in there, either more cells or cells in quicker? And there's not as much information as we would like regarding those things. So I want to talk a little bit about the process to get to transplantation. So if we find a patient that needs transplant, whether they have leukemia, whether they have Hurler or some other disorder, one of the big questions is, can we find a donor? So if you have a family that has several different kids, then the brothers or sisters could potentially be a donor. So the way that you inherit your tissue typing is similar to the way you inherit your disease like Hurler. It's a 1 in 4 chance that any brother or sister could potentially be a donor. Now, in a lot of circumstances, they're actually going to be carriers, which is a potential issue. And so it'd be optimal if we didn't use a carrier. So if we exclude the kids that don't have a brother or sister that it's a good match, or that are carriers, there aren't very many patients that can actually get treated with a brother or sister as a donor. So ordinarily, then, we're looking outside the family. There's a couple different directions to go there. The primary ones are unrelated donors. So these are adults that have been tissue typed before. The HLA types are available in a database, essentially. And then there's cord blood, which is blood that was from a placenta, and the HLA types have been identified there too, and those are stored and frozen away. So once we have the HLA typing, we can look at both of those things. So and I don't want to spend a lot of time talking about HLA typing, but there's several different antigens that we look at, several different pieces of the HLA that we look at. And essentially what we want to do is figure out how closely matched is a potential donor. So we can look at A, and we look at B, and we look at C, and we look at DR, and you get 2 copies of each of those. So then the terminology we use is how many antigens are matched out of that 8. So if we're looking at unrelated donors, we historically have looked at 8 different antigens. And you could say you're fully matched, or you're a 7 of 8 donor, or whatever. 7 of 8 donors we would consider using, but obviously the closer matched you are, the better off you are. The cord blood side is a little bit different. We don't have to match quite as closely with cord bloods, and so we have a little more opportunity for finding cords that are a little bit more mismatched, but it still works. So the process essentially is, if we find a patient that needs to get transplanted, it takes about 10 days to get the HLA typing back. And I use HLA typing and tissue typing kind of interchangeably, but it takes about 10 days to get that back. We then have that information, we give it to the database, which is the National Marrow Donor Programs, who we use, or NMDP. And they can plug in that HLA type to their database and find out, both for unrelated donors and on the cord blood side, what is available that's going to closely match. So we can do those searches simultaneously. The electronic part of the search just takes an hour or 2, and then you can get a sense of who's out there as potential donors that might work. The next step then is to confirm the tissue typing. So we would, for instance, if we're looking at a particular cord blood, we would ask that we get a sample of that cord blood, and we double check the HLA typing, which is what we call confirmatory typing. And that takes another 10 days or so. One of the key things, though, is if you go the cord blood route, these are all frozen down in banks and ready to go. So there really isn't much delay if we identify a cord blood and confirm that the tissue typing looks to be what we want. So then you can just proceed with the transplant. The unrelated donor situation is a little different, because these are people that are out in the community, and they may be, international people. So, if we're going to transplant somebody in Minnesota and the donor potentially is in Berlin, then, we have to work with them to get the donor cleared to make sure that they're going to be a decent donor. And one of the questions is whether or not the donor's got anything infectious or any other problems that might hurt whoever's going to be transplanted. And then we also want to make sure that the donor is healthy enough that if they go through the procedure, we don't hurt them, because they're going to do this out of the goodness of their heart, essentially. So they have to come in and be evaluated and have a physical exam done, and they have a lot of blood work done, and we have to work around their schedules because they're doing it, again, out of the goodness of their heart. So if we decide then a donor is an adequate donor and we want to pursue them, then we have to work with the donor to find a time where they could donate. And sometimes they have to take time off work or whatever. So again, you have to work around their schedule. So at the end of the day, it's a much longer process if you're using an unrelated donor as opposed to the cord blood piece. So it adds at least another month, if not 2 months, on top of the time to get to transplant. A little bit about cord blood transplantation. So people figured out that there's a lot of blood stem cells in placental blood. And so the first transplant that was done was back in '88. That was a brother-sister transplant. And then they started storing cord bloods in these banks and then utilizing them for people that weren't related. So one of the key things for us, if we're trying to move forward to transplant quickly, is the cord blood allows you to do that and get to transplant probably, as I said, a month or 6 weeks earlier than if you had to go with an unrelated donor. Also, generally speaking, there's less graft versus host disease, which is the donor cells reacting to the patient. And it's a major complication of transplant, and we see less of it with cord blood on a global scale. So most of the kids that we transplant for MPS disorders now get cord blood transplants. This is just to show the number of available donors in the unrelated donor pool. So there's almost 20 million people now that have been tissue typed and exist in this database that we can potentially draw on. The cord bloods there's much fewer cord bloods. But again, because you don't have to match them quite so closely, most of the patients that need to get transplanted, we can find a cord blood that would work. And this slide is just to show, so this isn't for the metabolic disease specifically, this is for pediatrics in general, but the orange part of the bar here is the cord blood utilization. So over time, starting back in about 2000, we started using cord bloods more and more, and it's really expanded since that time. So I want to talk a little bit about the MPS disorders and what makes sense to transplant. So for MPS 1, if we divide it into the patients that have Hurler, or severe disease, and those that have more attenuated disease, we consider transplant as really the standard of care for the Hurler kids. Because again, they have neurocognitive problems, and really the best way to treat that is through utilizing transplant. For the attenuated patients, we don't generally transplant them. There's obviously a lot of risk associated with transplant, so that's one of the big drawbacks of using transplant. And if you don't have to do it because people don't have neurologic disease, then that's the sense, is that you should use enzyme replacement therapy for the attenuated patients. Now, some of the patients with attenuated disease may still have some neurologic issues, and so that might be a possibility, but just globally, this is kind of what we're doing. Newborn screening is actually going to change things a bit, because they're going to figure out for kids that are picked up by newborn screening, some of them we're going to know have Hurler, some of them we're going to know have attenuated disease, and there's going to be some patients we don't really know what they're going to look like down the road. So that's a problem, because we still need to figure out how we're going to approach those patients. In terms of MPS 2 or Hunter, so generally we haven't transplanted in the US many Hunter patients. Some of the initial data that was done, or was generated from the transplant of Hunter patients, suggested that it didn't work very well. And I think I don't think it works as well as it does for Hurler. But a lot of the patients that were transplanted earlier and reported were relatively advanced. So if we can find patients that are relatively early, then maybe you have a better opportunity to intervene for the Hunter patients. So that's still a caveat here. In Japan, they have a lot of Hunter patients in Japan, and they've been transplanting patients there. And there's a fair amount of data that it is a reasonable approach, but it's not something we generally do in the US yet. So for Sanfilippo, transplant for whatever reason, doesn't work as well as it does for Hurler. So for Sanfilippo, again, we generally don't transplant those patients. Now, if you can identify patients that are very early, would there be an opportunity to change the course of the disease? Potentially, but it doesn't come up very often, and I don't think at Minnesota we've transplanted a Sanfilippo patient for over 20 years. So for Morquio, because again it doesn't have neurologic disease, there's no major advantage to using transplant. There is some sense, again from some of the Japanese data, that transplant works reasonably well, or works somewhat at least, in alleviating disease. But whether it works any better than enzyme therapy or not is still kind of unclear. And again, if they don't have neurologic issues, then it's hard to justify the risk of transplant. So for MPS 6, we used to do a fair amount of transplant for MPS 6, but again, if they don't have neurologic disease, almost all these patients now are just going on enzyme and staying on enzyme. So that's not, we haven't transplanted a patient with MPS 6 for some time now. And then MPS 7 is such an uncommon disease that doesn't come up very often. And now there are things that one can do for MPS 7 outside of transplant too. So having gone through all this, MPS 1 is kind of really the prototype for the MPS diseases in terms of transplant. So issues regarding success of transplant, and again, we're going to focus on Hurler. So there's a lot of disease-related problems associated with having this disease, as everybody in the audience knows. So as we're monitoring patients after transplant or with whatever intervention, we want to capture as much information as we can about how transplant or whatever you're doing is impacting virtually all of these things. And it's difficult because there's a lot of variation in patients. Some patients are older than others as they go to transplant. And so it makes the data harder to interpret than we would like. So this is from a review paper from Europe, but this is essentially the treatment algorithm for MPS 1. So if you have Hurler, which is kind of on the left side here, you're younger in age of diagnosis, you go on to get enzyme replacement therapy in the peri-transplant period, and then you go on to get transplant. So that's really been the course. If you have more attenuated disease, this is now down on the right side, you're a lot of times older when you're diagnosed, and you just go on enzyme and generally don't cross over to get transplant, although there are some disclaimers here that it might be appropriate in some circumstances. So as enzyme became available, there were some patients that decided they didn't want to do transplant. Some patients with Hurler, and the families said, we don't want to take the risk of transplant, we want to just go on enzyme. And so this is a publication that just recently came out comparing patients prior to any sort of intervention and those that were on enzyme and those that got transplanted. And you can kind of see what the results are here. So, I mean, historically, most of the patients with Hurler, before we had anything to treat them with, most of them died by the time they were 10. So survival is better with enzyme therapy, but it's not as good as it is with transplant. So one of the things that or changed the field a little bit as we've been doing transplants for 30 years, but now as enzyme became available about 15 years ago, there's opportunity now to do combination therapy. And so this is something that we've been interested in. And so a lot of the patients now that are initially diagnosed, we would consider putting on enzyme therapy at least up through the time of transplant. And so there's been some controversy in the field about whether or not this is a reasonable thing to do, whether it makes sense or whether it really helps. But I want to just talk about that for a second. One of the other larger institutions that treats patients prior to transplant with enzyme is Manchester in the UK. And so because they do things almost exactly the same way we do, we pooled our data, and this is the results of that. So all these kids were on enzyme prior to transplant. Manchester tends to stop around the time that the transplant happens. We go for another couple months after just to make sure that the donor cells are there. but overall survival is very good in this population of patients. Now, the functional outcomes, again, are hard, and so try to split them into a couple of different pieces. The neurologic one to start. So this is the neurocognitive trajectory of patients that have Hurler. So if you don't treat them over time, they lose ground cognitively. If you then look at how they're doing in association with transplant, there's a couple pieces of information I want to get across to people. So if you look here on the left side, 87% of patients in this large study had a normal IQ at the time they got transplanted. But if you look a year or two after transplant, there's only 27% of the patients that fall into that category. And if you look at the severely affected patients, only 16% of them were severe going into transplant, but 45% of them were severe after. So we see this decline in cognition around the time of transplant to a year or so out afterwards, and then it tends to be quite stable. But this is an issue that we have not resolved in terms of transplant, is stabilizing the disease from a neurologic standpoint as well as we would like. And if you look at the subgroups, the boys don't do as well as the girls. If you start out with a low IQ, then it's more of a problem. If you're older at the time of transplant, it's more of a problem. And we used to use radiation. TBI is total body radiation. We used to use that for transplant, but we don't do that much anymore. So Julie Isengart at Minnesota actually looked at some of these patients that had gotten enzyme replacement around the time of transplant and compared how much of a drop-off they have in cognition to the patients that hadn't gotten enzyme before. So it's a small study, there's only 10 patients in each group, but the take-home here was that we didn't see as much drop-off in loss of IQ points on the kids that were on enzyme therapy as the kids that hadn't gotten enzyme therapy, which is a little surprising because enzyme isn't supposed to get into the brain very well. So maybe around the time of transplant it gets in a little better, with the chemotherapy that we're using, for instance. I don't know. But I think this bears looking at in a larger context and with more patients, but it was of interest. So other functional outcomes, orthopedic ones are another key issue. So this is looking at growth of the kids that have Hurler after transplant. So about 10% of the kids were lower than the standard growth curve before transplant, but if you look 6-7 years out after transplant, 70% of them have fallen off the growth curve. So transplant does not treat the bones well enough to maintain growth. And if you look at that data closely, the older you are at the time of transplant, the more likely you are to fall off that growth curve. So this is a slide that's kind of a synopsis of the different types of surgeries that the kids have after transplant. So it's carpal tunnel surgery, straightening the backs, fixing the hips, straightening the knees, and then cord compression. So all these things, despite having gone through transplant, a lot of the Hurler kids experience. So they have to have these types of surgeries. So again, the point is that transplant probably helps the skeletal system, but does not fix it. It's not a cure by any stretch of the imagination. Now again, if you look at that data a little more closely, you can divide the kids into those that had more enzyme and those that had less enzyme after transplant. And if you have more enzyme, it's less likely you will need to have surgery than if you have less enzyme. So more enzyme is better. This is looking at cord compression surgery. If you look at that group as well and you divide them into the younger kids and the older kids, the younger kids are less likely to need surgery. The kids that are transplanted at a younger age are less likely to need this kind of surgery. So again, we're looking at cord compression here, but it's essentially the same for virtually all of these. So carpal tunnel is the same way. If you have more enzyme, it's less likely that you're going to need to have the surgery. If you're younger, it's less likely that you need to have the surgery as well. So enzyme levels make a difference, and age at transplant makes a difference. So there are other issues too that we follow in these patients. So this is what corneal clouding looks like. And sometimes patients that are transplanted don't really have this as much of an issue, but sometimes it's a big problem in terms of their vision. Now there's surgery for this too, so you can do a corneal transplant and replace that cloudy cornea with a new one that's not cloudy. But it's not something that this is not something we want to routinely have to do. So what can we do to help make transplant better? So this is something we talk about a lot. So if you can transplant these kids earlier, the data suggests that they will have better outcomes. So one of the things we can do there is the newborn screening. So there's 10 states that are now screening for MPS I. Been a couple picked up in Minnesota over the last year or so. But this is now going to allow us to identify kids that are a week old or 2 weeks old. And so we wouldn't probably transplant them until they were at least a few months old. But the vast majority of patients that we transplanted about 160 kids with Hurler at Minnesota. There's only 6 of them that have been transplanted less than 6 months of age, because they generally aren't picked up until they're later and have, until they're older and have some problems. So this has got great potential to change outcomes for the patients now that we can identify them early. So another thing that might make a difference, because we were talking about enzyme levels, is how do you boost the amount of enzyme that somebody's going to have after transplant? So, we've changed a lot in terms of how we do transplants over the last 30 years or so. They're much safer now. We have more, ability to get people through. But 30 years ago, if you were transplanted and all the cells after transplant were donor cells, and the same thing if happens now, if we transplant you and you're 100% donor, the outcomes are essentially the same. There's no way that we can increase the amount of enzyme that's delivered with the transplant. So all we can do is try to get people totally engrafted with the donor cells. But there's other ways to potentially boost enzyme. So one would be with gene therapy. So this is a mouse study, and I don't want to spend a lot of time talking about it, but there's rationale here that I think is important. So if you look at the Hurler mice, and you look at the amount of GAG or storage material that's present in some of the various tissues, and so that's the gray bars here. And then if you transplant them, that amount of storage material comes down, and that's the green bars. But one of the things you can do with gene therapy is you can have the cells make much more enzyme than they would otherwise make. So it's super normal amounts of enzyme. And at least in the mice, if you do that, if you do gene therapy and the new cells, the new blood cells are making a lot more enzyme than would otherwise make, you get better correction. And that's what the orange bars are here. So you can see the drop off from the gray to the green, which is the transplant effect, and then the additional drop off that you get with gene therapy. So again, this suggests that the more enzyme you can deliver, the better off you're going to be in terms of correction. There is some clinical data in regards to this too, and this is from the Manchester group as well. But if you look at urinary GAGs in Hurler patients that are on enzyme, which is on the right side, and you compare those patients to patients that were transplanted with carriers, so they're making half as much enzyme, you still see a decrease in the GAGs. And then if you look at kids that were transplanted without carriers, so with normal cells, that's on the left side. So you can see that the amount of GAGs that come down with these various amounts of enzyme that are delivered through your therapy. So this is biochemical information. It's not clinical outcomes, but you get the sense that the correction is better the more enzyme you deliver. So one of the key points here is gene therapy has the opportunity to be better than transplant or augment transplant, for instance. And gene therapy is started. So there were several discussions here about the Sangamo trials. which are open for MPS 1 and MPS 2. Those are treating adults really at this point, so it's not something they, at least for now, can offer to a newborn, for instance. The Regenix Bio group was talking about their gene therapy studies too, which is primarily giving virus into the spinal fluid and the cisterna magna, actually. There's a study that opened in Milan in the last 6 months or so, where they're actually taking the kids' own blood stem cells and engineering them to make enzyme and then giving the kids back their own cells, as opposed to having to give somebody else's, which has a lot of potential advantages, but there's no clinical data in terms of that now. I don't know if they've enrolled a patient yet. We're also interested in Minnesota about opening a trial using a different type of gene correction using a sleeping beauty transposon, which is the non-viral way of getting enzyme expressed. So that protocol may open in the next 6 months or a year. So gene therapy is going to be available. It's the various groups that it's going to be applicable to, though, kind of varies as to the approach. But this is going to expand from here. There's also companies that are developing, and I think Armagen may have had some presentations here too, but they've developed a second generation enzyme that might get into the spinal fluid better. So they're interested in looking at this too. In terms of combination therapy, we looked at giving enzyme after transplant to patients to see if it might help make them better. So this is kind of a pilot study. There's only 10 kids that we enrolled. But you had to be a couple years out after transplant. You had to have donor cells there. And we wanted the kids that were less than 14 because if we're going to see any benefit in the orthopedic area, if they're too old, you probably aren't likely to see that. So we just wanted to get a sense of whether we could potentially get a response in these patients. So essentially the way it was designed is we did an evaluation of the patients, and then they were on enzyme for 2 years, and we looked every 6 months to see how they were doing. So we wanted to get a sense of whether people tolerated that okay, or whether there was any problems. And then we wanted to look to see how they were doing. So this would be hard to read. So of the patients that we put on the study, 50% of them had had enzyme in that peri-transplant period again, so around the time of their transplant. The age of the kids at the time of transplant varied a bit from, well, quite a bit, from several months to almost 3 years. The age of enrollment, so the age when they started the enzyme therapy, was between 5 and 13 years. So 3 of those kids, when we looked to see if they had any antibody at the time they enrolled, had a very small amount of antibody. It was measurable, but it was very low. And 3 of the patients had a bit less than 100% donor cells. So one of the things we wanted to look at is whether people make antibody after transplant. So virtually all the Hurler kids, if they go on enzyme therapy, they almost all make antibody because it's a new foreign protein. In this circumstance, though, it's different because these kids now have enzyme producing cells in their blood, so they're actually making enzyme on their own. And we wanted to get an idea whether that was going to change in terms of they're going to develop antibody. So there are 3 kids in this 10 that we treated that did develop antibody, which is kind of surprising because one would have anticipated that that wasn't going to happen because, again, they're already making their own enzyme internally. So if we looked at the GAGs in the urine of these patients, so there's a couple points I want to make here. So if you look at all the patients, so the blue is kind of the baseline and then where they ended up at the end of the study. So they had a statistically significant drop in their urinary GAGs as a group. If you looked at the patients that had no antibody, it was even a bit more pronounced. But if you looked at the kids that had this high antibody titer, their GAGs actually went up. So, and this again was quite surprising to us, but what we took away from that is some of these patients clearly make antibody after transplant if they're exposed to enzyme, as in ERT enzyme. And so, and it may actually get in the way of the benefits that the people are getting with transplant, because the GAGs actually went up from where we started. So this is it's a bit concerning. And if, I know people keep kind of asking, would staying on enzyme after transplant potentially be helpful? If you're going to do that, I think it's pretty clear that you need to be followed closely to be able to to identify any antibody that's being produced that might be problematic. So one of the other things, and I don't have time to go through all the data here, but one of the things that was of interest too is that the two youngest patients on the study, in terms of growth, it looks like their growth actually got better. So it's hard to draw a lot of conclusions out of two patients. But again, it was the two youngest ones that seemed to have the best response. The rest of them seemed to be relatively parallel to what you'd anticipate a transplanted patient would look like. So just a summary from this study, and again, it's a 10-patient kind of pilot study, but all the patients had some improvement in some parameters in terms of orthopedic things, range of motion and those types of things. We just talked about the growth in these two patients. We did 6-minute walk tests to try to see how people would do, and we also put the kids on treadmills to see how far they'd walk, how fast they walk, and whatnot. And so most of the kids had some improvement in terms of what they could do. But again, the antibody piece ends up being important, and it's something that we would, again, if families are anticipating potentially trying enzyme after transplant, this is a key thing that we need to look at. So other modifications in terms of improving outcomes for transplant. So we're always thinking about ways that we can make transplant safer. One of the things, especially with cord blood transplant, if you get a placenta and you store the cells, you can only get so many cells out of a placenta, essentially. So, and it's pretty clear from the data that the more cells you can give to somebody in a transplant, the better you do. So there's been a lot of interest in trying to make more stem cells from a cord blood. So there's a company called Magenta that we've been working with that has an agent that can do this. So essentially, what you do is take a cord blood, you thaw it out. There's an agent that helps expand the number of cord blood stem cells that are there so you can give more cells. So we're talking 100-fold, 300-fold more cells than you can otherwise give. Now, the more cells you can give, the quicker people tend to recover after transplant, and so the opportunities for infections and some of these other things might be reduced. A study was done previously in some of the adults that had leukemia and other blood malignancies, essentially, to see how well this worked. So that white bar there is how fast it takes to recover your blood counts after the transplant. And then when they did this cord blood expansion, that's the red bar. So the number of days that it takes to recover your white blood cell count after transplant gets moved over almost 2 weeks. So that's really encouraging. So now we have a study open at Minnesota. We've got, I think, 5 kids enrolled. 4 of them were Hurler kids, actually, to see if it works in this population of patients too. So again, what we're hoping is that would happen as a result of this is we can incrementally make transplant safer. So just to summarize things, so doing studies in these rare diseases is difficult, and transplant centers don't do everything the same way by any stretch of the imagination. So if you're trying to compare data from one institution to another, it's difficult. If we can, and this is something we're working on as transplant centers, but if we can better combine our approaches and we do the same thing, I think it will help us get a better sense of how best to do these kinds of transplants. So in terms of the MPS diseases, especially Hurler, a couple of the things that we really need to make better is we need to better treat the brain and we need to better treat the bone. So those are really limitations that we have with our current approach. Newborn screening, I think, is really going to help. How much benefit it's going to end up being is going to be unclear for a while. So we'll have to get more kids that are picked up very early, transplanted very early, and follow them for a long period of time to see exactly how much difference it's going to make. But I'm pretty confident that it's going to help outcomes. There are new things that are in development, including gene therapy. How will some of these things be used in combination, or whether gene therapy is going to replace transplant or not? Time's going to tell. But it's great to potentially have other things to apply to these diseases, because historically we've been very limited in what we could use. So I'm going to stop there.
Thank you, Paul, for this very clear and excellent presentation. We have time for some questions. Please feel free to ask.
Sure. Do you have a microphone out there somewhere?
We have a microphone in the middle. Please come up.
Oh, yeah.
Mm-hmm.
Hi, my name is Claudina. I have a daughter who is MPS 1, who is now 15 and was transplanted without any use of enzyme prior to transplant. She had her transplant at Duke. And I'm curious as to whether you've seen any cases of far out transplant kids. She's now 14 years out of transplant, getting enzyme. I mean, her enzyme levels are normal. Her cognitive functions are normal IQ. Is there a benefit to this far out introducing enzyme, additional enzyme?
So she's, 13 or 14 now?
15.
15 now. Well, so what we've generally seen with the cognitive piece is after you get a year or two out after transplant, it's pretty stable. So, and giving more enzyme is probably, she's neurocognitively intact anyway, then there's, you can't probably make it better. But it seems unlikely that even if somebody's got an IQ of 75 or 80, adding on additional enzyme 3, 5, 10 years after transplant are going to make that any better. I think whatever problems you've had in your brain related to the disease are probably not going to improve. So some of the areas that might potentially get better, though, would be the musculoskeletal things. So, would range of motion get better? Would the kids just feel better and be able to do more things? And so those are the kinds of things we're thinking about in terms of quality of life and activity levels and those types of things. So, so it's hard to tell, but I mean, those would be the things that you'd be looking to see some improvement in if you were to think about adding another therapy on.
Okay, thank you.
Sure.
Any other question? So I would have two.
Okay.
One question is, as you spoke from the combination of transplant and ERT, do you think that the antibody development in ERT post-transplant could make any difference on the efficacy of an effective transplant?
Well, so there have been some studies done in terms, and again, it's 93 or 95% of the kids that have Hurler will make antibody pre-transplant. Generally, what's happened is people stopped giving enzyme therapy, early after transplant. And if you look for a year or two out, virtually all those antibodies go away. So, but no one's looked before if you reintroduce enzyme, whether you would make antibody outside of the small study that we did. Mm-hmm. So, and again, it suggests that there's some immunologic piece of that enzyme that actually stimulates the immune system, whereas the enzyme that's being produced by the cells themselves doesn't seem to do that. Mm-hmm. So exactly why that is hard to know.
Thank you. The other question I have is, I think you are the king of transplantation in Minnesota. You, um, we will hear another presentation, and I have already heard that they are going to do transplant again with MPS 4 children. So now the main thing is the brain. They don't have any cognitive problems. The problems are in the bone because it's a bone disease, I would say. And you just mentioned that you can't correct the bone disease in MPS 1. So I would be curious for your opinion.
Yeah, I mean, it's not curative. Transplant isn't curative in terms of the bone disease in MPS 1. So this gets a little, if you have several different therapies, it ends up getting a little bit complex as to how you make these decisions. So one consideration potentially might be cost. Transplant is expensive, but enzyme therapy is head and shoulders more expensive than transplant is. But we don't generally decide what treatment we're doing based on cost. The risk piece we've talked about, because it's certainly doing transplantation is a much higher risk procedure than staying on enzyme. So that's the primary reason that one chooses enzyme therapy over transplant. The other big question is, and the data is, I think, becoming a little more clear, but if you actually compare them, what works best? And so one of the slides I presented here suggested that if you have hurler kids and you treat them with enzyme alone, or if you treat them with transplant, at least the survival is better in the kids that are transplanted. And there's some sense that the GAGs are lower with transplant than they are with enzyme therapy and whatnot too. So what works better is also another consideration. So can you extend that to other MPS diseases and say, you know, we understand that transplant's a lot more risk for Mercurio than enzyme therapy is, but would it work better? And I don't think we have that data. But I think that's something that one at least needs to consider in the realm of this as well.
So would you think it's worth trying?
Sorry, what?
Would you think it is worth trying it?
Well, in particular circumstances, I don't think it's totally unreasonable to do that. I'm hedging, obviously, but if you had a brother or sister, for instance, that was well matched and not a carrier, and the patient was very young, would I consider doing it? I mean, I think I would, but we'd want to make sure that the family clearly understood all the risks that were associated with doing that. Now, if a patient is older, if they're 16 and they don't have good graft choices and stuff, then it's a totally different set of issues. I don't think in that circumstance it would make sense at all. So it kind of would be specific to the situation at hand.
Okay, thanks a lot. Thank you.
Sure.
So I would like to introduce our next speaker, my colleague from the International Network, Jenny Noble. Jenny is the mother of 2 children with mucolipidosis, and since the diagnosis of her 2 children, she has spent many years searching and advocating for innovative ways to manage the complications of mucolipidosis, which is much less understood than MPS. And through this advocacy, Jenny has built a strong international network, which is the ISMRD. And though she's not a doctor, she's involved in a publication, and I'm glad to have you here and to hear what you did.
Thank you. Bone disease in lysosomal diseases is a logistical nightmare, really, to be honest. And I'm actually going to be talking today about mucolipidosis in particular and the bone pain that these patients suffer from. And pimidronate, which was a therapy that we discovered by trial of, talking to people. So I'm going to talk about that and mucolipidosis in particular. How do I turn this over? So pimidronate is part of a group of drugs called bisphosphonates. Now, these drugs are used to treat people who have osteoporosis, particularly women who have had breast cancer and severe treatment for that. They end up with severe osteoporosis. The benefits of bisphosphonates are that it does improve bone density, and in mucolipidosis and other LSDs, it actually improves bone pain. We are aware that there are risks, such as fractured femurs and osteochondrosis of the jaw, and these symptoms are manageable. This slide was given to me by Thomas Brolke's lab, so I thank him for that. But I wanted to show just that, so the top bone picture is normal bone, and the one below is osteoporosis. And what that means is that the soft internal parts of the bone, or the trabecular and endosteal surfaces of the bone, become very porous and very, very weak. And in mucolipidosis, they lose at both these surfaces, but they also lose at the subperiosteal surface, and that's a little white layer above the trabecular and endosteal surfaces. And it is this kind of bone loss that causes the severe pain. So the drugs that are talked about in mucolipidosis are zoledronate, pimidronate, and fosamax. Zoledronate is the newer of the bisphosphonates, and pimidronate is an older drug, and we have chosen to use the older drug pimidronate. So I want to introduce you to Hayden and Sarah. These are my two. Hayden's 36 now, and Sarah's 32. They were the first patients in the world to do a case study on the use of pimidronate and to look at what was actually causing their bone pain. So back in the 1990s, early 1990s, or late 1990s, when we started this journey, the children were in chronic pain. Hayden was having constant chest infections. He hardly ever went to school. Transferring the children from chair to bed or chair to car was just a nightmare. Every time we touched them, they would cry. Putting shoes and socks on, showering them was terrible. And instead of being parents with hands with love, we were the parents with hands that hurt. It really wasn't a pleasant time in our lives. At that time, Hayden was spending a lot of time in bed. He was particularly unwell, and we were told that he was probably going to die. He wasn't going to make it to his next birthday. And I kept thinking that this is not good enough. There's got to be a problem to this pain. What is the problem? What's causing this? So why did we choose pimidronate? I was at an Australian MPS meeting, and I was listening to Ed Wraith talk about pain management, and he was going through a whole list of drugs. And I remember sitting there thinking, ticking them all off. I've tried that drug, it didn't work. And I was going through until we got to the bottom, and he mentioned bisphosphonates, and I thought, well, you know how you get a light bulb moment and you think, well, I wonder what that is. I wonder if this is an option for us. And so I called an around-the-table meeting with Ed, Professor David Salentz from Westmead Children's Hospital, the New Zealand metabolic nurse, and a few others, and we nutted it out. And I remember Ed's comment at the time saying, unless you've got bone disease, this is not going to work. And of course, my answer to him was, well, how do we know what mucolipidosis has got? We didn't know anything about bone disease at that time. The disease was too new. And so Professor Salent said, I want you to go home, turn around, come back to Sydney. I'll have Sarah for a week and we'll do a complete medical workup. And that's exactly what he did. She had every test under the sun to try and ascertain what was going on. And the day before we left to come back to New Zealand, he sat me down and he said, I actually don't want you to fly home. I'm really worried Sarah's femur is going to push through her pelvic bone. Her bones were the age of a 90-year-old lady. And he was very worried. Anyway, we talked about where to from here, and he said, well, I think we'd like to trial pamidronate with her. Now, they use that very successfully in osteogenesis imperfecta, children who have brittle bone disease. My dilemma was I had to come home and convince, one, my paediatrician, and two, the government, that it would need to be paid for. Unbeknown to me, my paediatrician was already investigating it and already doing the behind-the-scenes work. And so when I came home and said, well, this is what I'd like to trial with Sarah, and then later on Hayden, without a doubt, he didn't have a problem, and so we got started on pamidronate. There we go. It's just changed. Sorry. What surprised us was how quickly these kids responded. Within 2 weeks of having the first infusion, they were both saying that they were pain-free, and I thought, my gosh, how can that be so? But it was. And at 3 months, Sarah got out of her wheelchair and started walking, and at 6 months, she walked unaided. And then at 18 months of treatment, Hayden had a complete turnaround in his health. He was going to school, his chest infections disappeared, his whole demeanour had completely changed. And he said to me one day, Mum, I want to stand up, I want to show you I can walk. And I thought, well, actually, you can't. You're a paraplegic, so Hayden has other complications of his mucolipidosis. And he was absolutely adamant that he was going to show me that he could walk. So we went away and got Sarah's walking frame. And sure enough, he stood up and he walked 4 steps. I was totally shocked. This is not the kind of response that we expected. They stayed on treatment for 8 years. They were very carefully monitored. We did regular bloods, we did regular urine tests to look for bone markers to see what was going on. They had regular x-rays, regular DEXAs to check that we didn't have abnormal bone growth, to check that we weren't heading towards fractures. So the kids were monitored very, very carefully. And I think what surprised us was that they were happy, they were healthy, their whole demeanour just completely changed. I do have to mention this because with the quick response to therapy, we got an endocrinologist involved, and I think he was pretty surprised at the response in the kids as well. And so we did a bone biopsy in each of them after 1 month of treatment, and then we repeated it again at 12 months. And what we saw in the first biopsy confirmed what Westmead Children's had found, that the kids had lost severely at trabecular and endosteal and at subperiosteal. But at 12 months of treatment, we saw very good repair at trabecular and endosteal, but they were still losing at subperiosteal. And I must reiterate that when we went into this, we did not go into it to see any improvement in bone. We went into it to manage chronic pain that wasn't responding to conventional drugs. It became a really important case study, and so the hospital that I was at, Westmead Children's endocrinologist, a whole lot of us got together and we wrote the case study up and we published it in the Journal of Inherited Metabolic Diseases. It's also on the ISMRD website if anybody wants to download it, but it just became an incredibly important piece of work. And from there, we were asked to, we had to spread the word. We were asked to present at the Australian MPS conference and then at the International Lysosomal Conference in Paris in 2002. And I have to say, probably that was the scariest thing I'd ever done in my life. I was the only non-professional on the scientific stage. But after Grace and I did so Grace did the scientific part of it and I did the family part of it. And after the presentation, we were surrounded by families and doctors wanting to know more and wanting to know how to treat these patients who were living in chronic pain. 2 minutes. Okay. So I'm just going to quickly go through a few more slides that show other diseases that are using these drugs successfully. So Robert uses pamidronate, and Holly was using zoledronate. And Holly was using zoledronate because she's needle phobic. But the results of that particular drug are almost the same as pamidronate. And then Anna has used zoledronate, but so far at this point has not had as good a response as they would have liked, and the doctor's now looking at giving it to her more often. So zoledronate is 12 monthly, and they're looking at bringing it down to 6 monthly. And again, Anna is needle phobic. Spence has been on treatment since 2005, and it's improved his quality of life, and his doctor wouldn't know what to do with Spencer if pamidronate was not available. Sarah is our longest treated patient. She's been on for 15 years. Like Sarah and Hayden, she's trialled frequency, she's trialled dose regime, and what works for her is IV 4 weekly. And she said it feels like having pamidronate recharges her batteries. We have just recently put Hayden back on to pamidronate. He has reached osteopenia level again, and he is back in chronic pain. So I haven't yet seen any changes in him, but I'm hoping that we will see him happy and smiling again. So what are we doing now? ISMRD has sponsored a bone research project that is in Thomas Brolke's lab. Sandra Pohl is doing that. And we were able to do that because we gathered partners to help us raise the money and support that piece of work. It's a very important piece of work. They are now in year 2, and hopefully at the end of this year we will have the results of their 2-year project. We're actively encouraging doctors to gather the data of the patients that they're treating. We need to have more papers. We need to have more evidence on the use of pamidronate. And so my conclusions are that pamidronate reduces bone pain. It gets patients mobile again. It's less invasive compared to the surgeries that these kids have to go through. Side effects are manageable, and it improves quality of life for these patients. Thank you.
We are already a bit over time. Maybe you have a quick question? So if not, I would like to introduce our next speaker. Dr. Fowler, are you here? Okay, fine. Dr. Alan Fowler is from the Georgetown University, from the US. He does research for potential drugs for Parkinson and Alzheimer's disease, for example, in preclinical models. And he will speak now about the efficacy of TKIs to reduce heparan sulfate via autophagy. Thank you.
Thank you. So I'm actually only still a PhD candidate right now, and I'm a Parkinson's researcher first, and then today I'm going to introduce some preliminary data on a project that my lab has been working on about the efficacy of TKIs to reduce heparan sulfate via autophagy. So what is autophagy? Autophagy is a quality control mechanism that every cell has to degrade waste. And so under normal conditions, waste in a cell can be degraded via two major autophagic pathways, the first being the ubiquitin proteasome pathway. And so if waste were to go through this pathway, it first needs to be tagged with a ubiquitin that allows it to be recognized by the proteasome, where it can be recycled into individual amino acids. And this pathway can be thought of like a carry-on luggage for a flight. So the piece of luggage has to be very specific dimensions for it to be accepted into the overhead compartment. So if a piece of waste is too large or misshapen, it won't be accepted into the proteasome for recycling, and it'll have to follow another pathway to be degraded. And that alternative pathway is called the autophagy lysosome pathway. And this is a less specific bulk degradation waste removal system. So now waste going through this pathway, there's an initiation of this pathway by two proteins, one called Beclin and the other called Parkin, that begin to sequester the waste inside of a new structure called a phagophore. That phagophore then matures into a complete double membrane-bound structure, which then fuses with the lysosome and deposits the waste into the lysosome for degradation. So one area where this system has been exploited as a therapeutic treatment is in cancer. So the underlying problem with cancers is that you have cells that are abnormally programmed to continually multiply and form masses or tumors. And so what's happened is tyrosine kinase inhibitors have been utilized in cancers as a maintenance therapy. So if a mass or a tumor is removed with either radiation or chemotherapy, a patient is put on a tyrosine kinase inhibitor to then keep the cells from multiplying back. And how that works is that if this system is overstimulated, but there are no waste species to be degraded, it forces the cells to degrade essential organelles and structures and essentially self-cannibalize. So in cancer, it's utilized to prevent cells from multiplying back by continually forcing the system on and causing the cell to cannibalize. Now, what my lab has done is pioneered the strategy of using tyrosine kinase inhibitors for neurodegenerative diseases, where a protein is the waste species that accumulates inside of the cell. So in neurodegeneration, if you follow the red arrow to the left, we've found that this autophagy lysosome system is also dysfunctional. So when it's turned off in Parkinson's or Alzheimer's disease, you have neurotoxic proteins that are the trash that builds up, become neurotoxic, and lead to cell death. And then for cancers, as a therapeutic strategy, this system is overstimulated. It's always turned on with very high frequent doses of the tyrosine kinase inhibitors. It will degrade any trash or accumulated species and then force the cell to degrade essential organelles and also lead to cell death. And so an example is nilotinib, which is a tyrosine kinase inhibitor that has been approved for pediatric CML, which is a type of leukemia. And what we've done is we've adopted that therapeutic strategy towards neurodegeneration by administering lower, less frequent doses. So we turn on this system for a few hours a day in a neurodegenerative disease. And that allows us to degrade some of those neurotoxic proteins that have built up inside the cell, but not force the cell to degrade essential organelles and lead to cell death. And so this strategy promotes cell survival in those patients with a neurodegenerative disease. And so now, what if the species of waste that's building up is no longer a neurotoxic protein? So the therapy is essentially not non-protein specific, which has allowed us to apply it to many different neurodegenerative diseases, Parkinson's, Alzheimer's, ALS. But because it's not protein specific, it may not be substrate specific either. So now we are beginning to investigate whether we can apply it towards MPS 3, where the substrate or the waste is heparan sulfate. And so our aim is to investigate whether we can stimulate the flux of autophagy with tyrosine kinase inhibitors to degrade accumulating heparan sulfate in an MPS 3A animal model. So what we did is acquire the MPS 3A mouse model, which mimics Sanfilippo syndrome. It has reduced sulfamidase activity within the lysosomes due to a mutation in the SG-SH gene that encodes for sulfamidase. And these mice are generally characterized by accumulation of heparan sulfate in the lysosomes of all of their organs. And so we treat these animals with two different types of tyrosine kinase inhibitors, one being nilotinib, which is already FDA approved for the pediatric CML, and that we have moved into clinical trials for neurodegenerative disorders, and then another tyrosine kinase inhibitor called bosutinib. And we look at outcomes such as behavior, we test before and after treatment, and then we look at the heparan sulfate and the proteins related to autophagy lysosome pathway. So here is some rotarod data. So rotarod is one behavioral test that we use in the mice, where the mice are placed on a spinning rod. It's essentially a treadmill, and it's testing for balance and strength and speed. And so we measure their ability to stay on this accelerating treadmill over a 5-minute period. And we report the time that it takes for them to fall off, or the latency to fall. And so if we look at first on the left, wild-type normal mice compared to the MPS 3A mouse, we see that the MPS mice have a significantly lower performance on this task. And then with 21 days of treatment on nilotinib and bosutinib, the post-treatment data shows that the nilotinib-treated animals are no longer significantly weaker on their performance on the rotarod task compared to a normal mouse. The MPS DMSO treatment, which is essentially no treatment, and the MPS mice treated with bosutinib still have a significantly weaker performance. And then we can look at individual animal performance on the right. And we see that there's a trend if we look at the center of the graph at just the animals treated with nilotinib, that the animals that were more severely affected before treatment had more improvement after treatment. And then we confirm that the levels of heparan sulfate in these animals. So we measure the amount of heparan sulfate in the brains of these animals, and we see that the MPS untreated DMSO animals have significantly higher levels of heparan sulfate compared to a normal animal. And then when we look at the two animal groups that were treated either with nilotinib or bosutinib, we see that those animals have had their heparan sulfate levels significantly reduced after the 3 weeks of treatment. And then we can take the brains of those animals and section them and use fluorescent staining to look at LAMP2, which is a marker for the lysosome, and heparan sulfate. And so if we look at the untreated animals on the left, we see that the LAMP2, or the marker for the lysosome, is very faint and doesn't have a distinct structure. And then if we look at the heparan sulfate in these animals, we see that it's fragmented in long chains and scattered. And when we overlay these images, we see that there's no co-localization. So the bottom image shows that the faint LAMP2 staining for the lysosome doesn't match up with any of the heparan sulfate, which leads us to believe that the lysosomes are not intact and that the heparan sulfate is now spilling out into the rest of the cell. When we compare this to the animals that received the tyrosine kinase inhibitors, nilotinib and bosutinib, we see now that the LAMP2 staining is brighter and it has a clear, distinct structure. And then when we look at the heparan sulfate in these animals, we see that those also have a distinct structure. And when we overlay these two images, we see that they co-localize. So now all of the yellow is where the LAMP2 staining and the heparan sulfate staining are together. And that leads us to believe that the lysosomes in the treated animals are intact and that the heparan sulfate is isolated or sequestered inside of those lysosomes. And so as a neuroscience researcher, my main goal with this project would be to reduce the level of heparan sulfate in the brains and alleviate some of those cognitive and motor symptoms that may be related to neuronal dysfunction. So only in the last few years has it been found out that MPS 3 patients have some of the neurotoxic proteins accumulating in their brains that you would see in a neurodegenerative disorder. So alpha-synuclein we know is associated with motor dysfunction and Parkinson's disease. And then tau and amyloid beta are associated with cognitive dysfunction in Alzheimer's and other related dementias. And so when we look at the brains of these MPS 3A mice, we see that alpha-synuclein, phosphorylated tau, and amyloid beta 42, which are all neurotoxic versions of those three proteins, are significantly elevated in the MPS mice which did not receive treatment. And then with treatment of the nilotinib or bosutinib, we see that the level of those three neurotoxic proteins are significantly reduced. And so our running hypothesis now, and what we've seen from our experiments, is that heparan sulfate is accumulating inside the lysosomes of these MPS 3A mice brains, and there's an accumulation of neurotoxic proteins that are not being degraded by the lysosomes. And when we looked at that fluorescent staining, it looks like the lysosomes are becoming fragmented, are bursting, and the heparan sulfate that could have been isolated within them is now spilling out into the rest of the cell. And then when we treat these animals with the tyrosine kinase inhibitors, we see new or higher numbers of intact lysosomes. And so we think we're inducing generation of new lysosomes, and those new lysosomes are now providing more of the other three non-mutated enzymes that are helping to start to degrade some of the heparan sulfate that is accumulating. But those new lysosomes have also not become fragmented from heparan sulfate overload and are able to help degrade some of the neurotoxic proteins that are beginning to accumulate as well. And so in conclusion, the SGSH mutation leads to an inability of the sulfamidase to degrade the heparan sulfate, and the other three proteins alone are not enough. The heparan sulfate is accumulating inside the lysosomes, which then become overwhelmed, fragmented, burst, and leak the heparan sulfate out, which we saw in the fluorescent staining. And then neurotoxic proteins are beginning to accumulate and have no available lysosomes for protein degradation, which leads to cell death in the brains of these MPS 3A mice. With tyrosine kinase inhibition, we induce Becklin-mediated autophagy and stimulate the overall flux of the autophagy lysosome system, induce lysosome generation, which provides more of the other three non-mutated enzymes that are available to degrade heparan sulfate. We begin to clear the heparan sulfate, and we also begin to clear some of the neurotoxic proteins which begin to accumulate, promoting cell survival in these animals. So I'd like to thank my thesis mentor, Dr. Charlie Musa, and then the Laboratory for Dementia and Parkinsonism and the Translational Neurotherapeutics Program, which is where I do my research, and then the other members of my lab that are assisting with this project, and the National MPS Society for funding this preliminary data and future studies.
Thank you for this very interesting talk and the nice data you have presented.
Thank you.
Sounds very promising. Is there any question from the audience? Please go forward.
Grzegorz Węgrzyn from University of Gdańsk. I have one question, one comment. So the question is, how was the drug administered to mice?
IP.
IP, okay.
Yes, we originally tested both oral gavage and intraperitoneal injection in the animal models, but they have roughly the same efficacy in the animals, the same brain penetrance, both methods of administration, but for animal models, it's just easier to give injections than it is to orally gavage every animal.
Okay, thank you. And a comment, because we tried a very similar approach, but using the natural stimulators of autophagy.
Okay.
And we found that really this, with different MPS3 subtypes, fibroblasts, they work. I mean, they decrease the GAGs efficiently. So we can see a poster 80.
In cells.
So yeah, so it's, it can support your idea that autophagy stimulation may be the way to decrease the storage.
Right.
Okay.
Yes. Thank you.
Thank you.
So thank you very much. Now it's my pleasure to ask Shunji Tomatsu, my special friend, to give his presentation on more care updates, for example, on care, trachea care, even on trachea surgery, and some other advanced therapies. Please go ahead.
Thank you very much. I want to talk about molecule A syndrome. So you can see many faces of a molecule, wonderful people, but each patient has a different unique feature, face and character, sometimes smiling, sometimes very sad, but like, other ordinary people. But it's quite a variety of, clinical phenotype. That's, you must say that. We just, this is not advertised, but we are just publishing, it says, it's called the Mucopolysaccharides Update. Christian Lovely actually dedicated this textbook a lot from UK Society. So I just want to mention, this textbook is, again, supported by UK MP Society. So because of that, really family-oriented. And also, we have received many masterpieces from, in this case, I think, molecule patients. I think it's Maria? Yes, so it's a masterpiece from Maria as well. So anyway, so this is really family-oriented kind of chapter book. If you have an interest, please let me know, okay? So I'm going to talk about molecule A therapy update. So we understand that there are many therapies. So major talk today is that tracheal reconstructive surgery and current available to treatment, ERT and hematopoietic stem transplantation, and small molecule gene therapy and substrate deduction enzyme therapy. So tracheal reconstructive surgery, it's began since 2015. It's very brand new, surgical procedure, which is maybe applicable to other type of MPS as well. To give the credit to Dr. Pizarro, cardiac surgeon at our hospital, started this surgical procedure. Because tracheal obstruction is a major life-threatening issue for many of molecule patients. Sleep apnea, respiratory failure, and how often, how severe, how we solve this tracheal obstruction. Major cause of death after teenager, okay? And also, if we have tracheal obstruction, anesthetic procedure is not so easy. Intubation, extubation, quite a trouble. So this is I'm schematically showing how it happened. Most important thing is that for molecule is that imbalance of growth. What's the meaning of imbalance of growth is that, in general, healthy, people, straight trachea. However, majority of molecule patients in teenager, above teenager, like twisted, and more likely, redundant trachea. So spine stop growing very fast, which means short neck, okay? However, trachea is still growing in particularly molecule patients. That's causing a lot of trouble, which is narrowing trachea will happen. So this is a typical, narrowing trachea here. You can take a picture as much as possible. I don't care, okay? All right, this is a family-oriented, slides. So again, this one is a typical CT tracheal obstruction here. You can see that twisted, kind of 80 to 90% obstruction happening here. So this new tracheal reconstruct surgery has happened again 2015. 16 years old boy, patient under 2.5 years of enzyme repair therapy, getting worse and worse about breathing difficulty. And they're starting to use BiPAP, ventilation. So finally, diagnosed with a severe obstructive lung disease, CT scan and MRI demonstrate a compression of the anterior trachea by it's called innominate artery. Okay, so his parents decided is use due to concerns about lifestyle and the challenge associated with maintenance. Because of that, they diffused tracheostomy. Actually, a very famous university and other university recommended a tracheostomy, but the parents diffused because the tracheostomy is a lifestyle quite changed. So they finally knocked our surgeon, Dr. Pizarro, as I showed you. So he decided to do tracheal reconstructive surgery, okay? Because that is the most important part. So this slide I just received from the family directly. I just want to show you. My innominate artery bended over my trachea here, which was flattened to 70%. So around 50 years old, I can't, when puberty, is that correct? Yeah. So whatever. Anyway, he needed, like Darth Vader, noisy kind of, voice, and cannot breathe, and even he's using BiPAP. That was a situation, getting worse and worse. So he knocked our door finally, and then what happened is that you can see that it looks like paper thin slice, like a trachea. You can see that. So this is why he cannot breathe. That's a anatomical issue because of compressed by artery here in front of trachea. Okay, so trachea is artery is over the top of trachea, so it's compressing here. So you can see that. So what type of surgery Dr. Pizarro and his group did? So you can see that trachea on top, artery is here. So this artery is compressing trachea. So first of all, innominated artery relocated like this. Okay, so not bothering trachea anymore. And then after that, since trachea is redundant, it's quite long compared with the spine. So what he did is that cutting several trachea and then put together. That's what he did. So now trachea is straightforward and then again inflated. So that's a situation inflated. Okay, and we checked post surgery. We got the tracheal specimen. What we looked at here is that full of storage material in trachea, which means, as we expected, enzyme cannot penetrate bone and cartilage. That's what's showing here. So this is a tracheal surgery, 1 week hospitalized, day one. You can see that. Actually, this one received by the family as well. Day one post-surgery with a ventilator, a stable and tolerating ventilator. Day second, you can see that ventilator was removed already, and the new trachea was put into the test. My trachea tissue was not floppy. It was very hard, actually. Not floppy, actually, trachea itself, which means anatomically, we can say that. And the trachea holds wonderfully. That's quite promising. Day second. Day third, post-surgery. Transition from, he doesn't need, O2 mask now, and no nose cannula here. Now, much easier to move. And then day four, breathing ambient oxygen well tolerated. Here, much better. He can movable now post-surgery. Oxygen is out. And he can write, sit down, now reading, possible. And he can eat already yummy breakfast, work with PT, and working on the computer. Day five, more active. Day six, physical therapy is now starting. And day six, walk alone. And day six, more yummy breakfast nutrition. So getting more energy. So then day seven, already discharged. Isn't it impressed? Yeah, very impressed. Yeah. I'm surprised as well. And then 1 week post-surgery, already touring around with my family, Washington, D.C., Philadelphia nearby. And then 3 months later, what he could do that post-surgery, drumming, swimming, and canoeing, actually paddling, kayak. So he could fully active after this surgery within 3 months. That's amazing. And the mechanism of trachea obstruction, most important part is that imbalance of growth, for sure, for molecule here. And also short neck. And also innominate artery, as I told you, large mandibular or pectus carinatum, pigeon chest as well. Okay, there are multi-factors to cause tracheal obstruction. And so far, we have 10 cases, and 4 institutes successfully received this new surgical procedure. All are above 13 years old. So age is a very important factor. So 8 cases were under enzyme replacement therapy, 6 months to 5 years. Before surgery, respiratory status is that we call it look up the sky position. Some molecule patients walking like this. That's a kind of sign of tracheal obstruction. Okay, loud breathing, loud snoring, shortness of breath, BiPAP or CPAP, whatever. And post-surgery, easy breathe, no look up the sky position, no snoring, no artificial ventilation, marked improvement of activity of daily living. That happened. So message prior to any surgical procedure. Of course, this is not low-risk surgery. We have to say, molecule patient with a teenager is recommended to take MRI, CT, angio for trachea to check first, okay? And then, of course, if sign and symptom, as I told, suspect tracheal obstructive airway, definitely, we need annual check for sure. So well informed, we're prepared for surgical procedure, okay? And the next step is that why it's so difficult to reach the bone. Big reason is that, especially cartilage, okay, growth plate is avascular. So it's hard to deliver the drug to this region. That's a fundamental problem. So enzyme replacement therapy currently available, but that's already a limitation of current ERT for a bone lesion in MPS 4A, no improvement of bone pathology in mouse model, no proof of concept to reduce bone lesion at this moment. We checked over 20 and our surgical remnant after 6 to 16 months, 5 to 24 years old surgical remnant, no clearance of storage material in chondrocyte. Still, we need surgical intervention, whatever surgery is. And another issue is that that already urinary case is reduced, but after enzyme replacement therapy, urinary case does not correlate with clinical improvement. If 70%, 80% of reduction is happened, but you don't feel such clinical improvement because blood case has not reduced in parallel. That's already we published recently. And half-life time in circulation, 2 minutes in mouse, 14 minutes in human. So which means circulation time is very short. So because of that, it's hard to reach the bone. Okay, that's we know already. And now we are switching gear to new type of therapy. So we are collaborating with Spanish group, development of enzyme therapy with novel nanomedicine. I just want to briefly introduce. So since enzyme cannot prolong circulated, so because of that, encapsulated by nanomedicine, and then long circulation will happen. So the reaching the bone. That's a kind of strategy. So they finished now in vitro data, and we are next stage that we would like to do that actually. This is part of pathology, tissue with formation. You can see that nanoparticle here and there, and this is a deposit gag. So if nanoparticle is inside the cell, they can digest, enzyme can digest more efficiently. That's we will conduct on our MPS for a mouse model. So future perspective, definitely we need still in vivo study on mouse model. So how to solve the bone lesion again? Important thing is that keep enzyme activity in blood circulation as long as possible, deliver the enzyme to target the tissue, which means bone, or early treatment helps. So another point is that small molecule. This is coming from Colombia, Javier's group, which I have been associated with him for many years. So he is searching a small molecule, okay? And small molecule means molecule itself is such a small size, can be, can get into the bone. That's a possibility. So that's another type of drug, which is we have to think about, okay? So it's called chaperone therapy. I don't want to tell you in detail. It's not, required at this moment. Important thing is that chaperone therapy, small molecule, can have a possibility to penetrate to the bone, okay, and stabilize the enzyme. That's the mutant protein which a patient has. That's a theory. It works. So already his group done about in vitro data. Quite encouraged. I don't want to show. I don't want to talk in detail. But the important thing is that we should move together to in vitro study, which means by using a mouse model. That's also another encouraging new therapy. Okay, I want to switch back about the cell type of therapy now. Okay, one is that currently conducted in clinical practice, HSCT. So this is a Japanese boy which I have known many years. 5 years like this, in the 8 years completely wheelchair bound, 15 years already completely bound. But after this HSCT, he could now stand up and he could walk, okay? And can you, hey, could you please do that video? Both patients together. Thank you. So this is a sorry, one more time, I'm sorry. So 15 years old boy got HSCT, and now 26 years, 11 years after HSCT, he could walk. Okay, and this is more early stage. A 4 years old girl got hematopoietic stem transplantation. She is now 25 years old. She looks like normal walking, as you see that. There is no limitation to walk, and she can squat easily, and raise the hand quite easily. Okay, again, already 21 years has passed after HSCT. That's amazing. This is a best patient at the age of 25 years old with severe type of MPS, which I met. I met over 400 MPS 4A patients. But this is a good example how cell therapy will work. Okay, so she is a full employee, and she has no limitation about her working ability. Okay. So result about HSCT here, I didn't know that actually. Recently I noticed This HSCT project was approved actually in '95 in Japan. That's happened already at that time. And since, Japanese transplant doctor published this data recently, 2014, I didn't know that this one as well. In China, many molecule patients are now transplanted, okay? There is a big team now at Shanghai. So they are doing 9 patients of transplanted, okay? Like this here, year by year. And what they found is that all alive, so successful, and achieved through donor chimerism, and the enzyme level is normal, no severe complication, significant improvement in the ligament laxity. That's we didn't expect, but looks like. That's what's showing, okay? We need, of course, long-term more efficacy, therapeutic efficacy by HSCT. Okay, switching gear to now gene therapy, okay, which we are now doing at the lab level. So maybe already you heard about gene therapy a lot during the conference here. So we are doing in vivo gene therapy here, so direct gene therapy by using, it's called adeno-associated virus. Okay, also another uniqueness is that we used liver targeting adeno-associated virus gene therapy, which means enzyme is produced in liver. So we have 2 construct gene vector. One is wild type. Second one is bone targeting gene therapy. Okay, so biomarker is that enzyme activity and the keratan sulfate level in blood. Okay, again, we have to measure blood chaos. This is very important. So blood enzyme activity here. So over the time of the course, this is a very unique. 4 weeks at a mouse model, we treated by gene therapy. So 2 weeks later, already 20 times, 40 times, above normal level. So enzyme is produced, tons of enzyme is produced, blood circulation. Even slowly better, higher after we sacrifice the mice. Okay, we followed 16 weeks old. Okay, and then result, enzyme activity is kept as long as possible. That's we encouraged. So then, how about enzyme activity in liver? Since this is liver targeting gene therapy. So what we see that here, it's very interesting. Wild type is just 6, but both vector, bone targeting native vector is quite a higher liver enzyme activity. That's showing here. So result is also again encouraging. Liver enzyme activity is much higher than actually wild type level. Actually, we already assayed the keratan sulfate level in liver, completely normalized keratan sulfate in liver. And then keratan sulfate level. Okay, this is a kind of storage material, a major storage material in molecule. So this is another surprising. We started treating mice 4 weeks old. Just 2 weeks later, keratan sulfate in blood completely normalized. This is fantastic. We didn't expect. Okay, and this is untreated. This is a treated mouse. Completely normalized until we sacrifice at 16 weeks. Okay. In previous study, okay, knockout mouse model were treated by enzyme repair therapy. Ks level in the plasma blood, which means were not normalized even after 12 weeks infusion. Okay, we already published this data, which means gene therapy is more providing impact. Pathology in bone. So I skipped this one. So focusing on first femur articular cartilage. So if you can see here, this is untreated and native type, and this is bone targeting. So maybe it's hard to say something, but only we can say that it's completely normalized by bone targeting, more near normal. Okay. Also, native one is much better than good structure, column structure compared with untreated one. Same thing through growth plate. it's more likely, regular pattern and near normalized in growth plate region by using, human gallons native type or bone targeting is better. Okay. So heart valve, switching gear. Why we have a concern about heart valve? Because 1/3 of patients, molecule of patients, died of heart disease. So we have to check, heart disease as well. This is very important. So what we look at here is that, okay, this is untreated. A lot of, storage material here along the heart valve base part. But both treated mice, native or bone targeting, nothing. Completely limited, no vacuolated cells. Okay. Okay. And I'll skip this one. Conclusion is that gene therapy is a very promising result. And this one is supported by Regenxx Bio. I thank you to them. And one more treatment which we are conducting, the substrate degradation enzyme therapy. This is a thermostable keratinase, inexpensive and stable in circulation. What the difference is that this enzyme works at the neutral pH, not acidic pH. So it's not necessary to go into lysosome. Blood circulation or extracellular matrix, it works. So we treated a mouse model several times. So again, what we surprised is that just one-time injection, blood KS level is completely normalized. So it's maintained, up to 12 weeks. But, we did it like several weeks, infusion. Okay, not every week. Okay, like every 4 weeks. So that's even though it's effective. So less frequent infusion. So bone pathology, you can see that this is normal. This is untreated, and compared to untreated, it's much better column structure and less storage material. So conclusion is that another treatment, thermostable keratinase, reduces the accumulation of KS in blood and bone tissue. That's you can say that. Okay, this is provided, supported by Seika Corporation, Japan. I appreciate that. So message here, this is the last slide. What kind of treatment is the best? Of course, our goal is that bone impact. Less side effect, for sure. Safety is a very concern. Possibly less frequent. One time is the best, but possibly less frequent. And of course, in future, less surgical intervention is better and less expensive and make a risk and benefit. Thank you very much.
Very encouraging presentation. We have not got enough time for questions, but I would suggest that we take the break for any questions you might have to Shunji or to Paul Ochert, who is still here with me. Would you like to ask questions? Please.
That's a good question. Actually, I cannot answer that part. Maybe you should ask the company because that company is, leading, clinical trial for sure. We can do that like this mouse experiment, other type of animal experiment, fine, as much as possible. But clinical trial is different, totally. Company should lead. Excuse me? Bone transplantation, like, bone transplant. Bone marrow, sorry, bone marrow transplantation. Yes, something like that.
So I have a question. I don't know if this works. So if you're looking at the trachea, thank you. So if you're looking at the trachea, what's the best way to identify the abnormalities? By CT or by
CT angio. CT
by actually
angio.
looking?
Yeah,
Okay.
yeah, CT angio is the best.
And so, and if you need to repair the trachea, in what proportion of the cases are you going to be able to do that with just cutting some of the abnormality out and putting it back together, as opposed to putting in another graft, for instance?
No,
or
no, we
put-
don't do that graft.
Oh,
Just cutting redundant part.
Okay.
Then
So,
disconnect.
so in all cases, there's enough normal trachea there that you can just cut out the abnormal
Yes,
piece and
yes, yes.
re-anast.
Yes. I'm talking about very simply, but it's quite a high risk surgery again. I don't want to recommend general hospital will do. No.
So I think, Shunji, you will be around for other questions.
Yeah, I welcome any question on the floor. Yeah.
You might need
Yeah.
need to refresh yourself. We have maybe 5 minutes. Please come back to 3 o'clock, and I would like to change my chair with Carmen. Thank you very much for all the presentations.